Okay, this talk is on DNA mutations, and I'm going to start with the definition, and I'm going to go through a couple of example mutations. So DNA mutations are any change in the nucleotide sequence of the DNA. So for example, that's any of the A's, the T's, the C's, and the G's. If any of those get changed, we consider it a mutation. Some causes for this. The first one is just randomness. Anytime the DNA is copied, there's a random chance a letter gets mixed up. Radiation can cause this, so sunlight, X-rays, etc. And chemical mutagens, so tobacco, alcohol, those types of products. Some good things that can result from mutations. One is, for example, eye color. We previously, as humans, we used to always have brown eyes, but now we have blue eyes as a result of a mutation. Something that's a little bit more applicable to our life is lactose tolerance. So in early humans used to only be able to drink milk, which has lactose in it, as babies. And then they would grow out of that tolerance level. Now we have lactose tolerance in adults, and that's a result of a mutation as well. So those are a couple of good things that have occurred. Several bad things occur also with mutations. Now, not all things are bad, but the diseases, sickle cell anemia or Tay-Sachs disease, are two example bad things that occur in mutations. The first example I'm going to go through is a silent mutation. Here's an original DNA strand, so consider it ATG, CCA, TCG. Now in the case of a silent mutation, this A gets switched to a T, for example. So we have, in our mutated DNA, it's ATG, CCT, TCG. So when you go through the RNA process to an amino acid, we take our original protein, so that's this strand up here. Our original protein's MET-Pro-Cir. Our mutated protein would be MET-Pro-Cir. So even though this letter changed, it didn't change the amino acid here. So there's no change in the amino acid in a silent mutation. Another point mutation we have is called a missense mutation. So in this case, this middle C gets converted to an A, again, probably in the DNA replication process. So original protein, if it was taken from this sequence, would be MET-Pro-Cir. And the mutated protein would be MET, and then glycine, Cir. So we have one letter change now that actually changed, causes one change in the amino acid in the protein. Our next one is called a frameshift mutation, and it's an insertion or an inserted letter. So we take our original DNA strand, and an A gets inserted right here before the C. So our mutated strand is ATG, ACC, ATC, and G, which is quite different than our original one. Our original protein would still be MET-Pro-Cir, just like it has been, but our mutated protein would be MET-Ala-Yl for the amino acid sequence. So by this A getting inserted here, it changes everything down the line. So this changes any amino acids after the inserted letter. So after the letter A gets inserted there. The next one is a frameshift mutation called a deletion. So if we take our original DNA strand again and we delete the letter C, we get ATG, CAT, CG, question mark. So our original protein in this case would be MET-Pro-Cir. Our mutated protein would be MET-Lu, question mark. And the question mark comes from because we don't know necessarily what's over here past the G. So we don't know what that letter is, so we won't know what that amino acid is because we don't know the third letter. So this changes again every amino acid after the letter is deleted. So after we delete that C, everything shifts down this way one spot. Last thing here, as a review, mutations can be good or bad. Good things about mutations is they often develop new traits.